Hey guys, okay, so I had to take a little bit of a break there. Funny it is, call my insurance company to figure out why my teledoc services were down. But anyway, I have to wait a couple business days. Life as it is, right? Okay, so hopefully you guys are enjoying your Friday. And you saw some Olympic coverage yesterday. I was so passed out. I just had to like wake up early in the morning and then figure it all out again. So enjoy your Friday as much as possible and your weekend, but kind of enjoy your studies as well. Those guys are just stories about really understanding meiosis and mitosis in some detail. And hopefully, especially with these long PowerPoints, you guys get this as a story as opposed to just a series of facts kind of thrown at you, which really doesn't do anything, okay? All right, so let's talk a little bit about alterations of chromosome number and structure and how that happens, especially in the body. Remember, we have 23 pair, 46 total, and these guys are going to continuously divide. If you've got meiosis 1 and meiosis 2 with your gonads for your ovaries and testes, what you're going to see is after the first phase of meiosis 1, meiosis 2, those guys are just going to undergo sexual reproduction through mitosis, right? Once it's finished with that. So there's a couple of things we should take a look at is accidents. Accidents actually can change the number of chromosomes. So we can see how mutations happen in the body, right? So there's something called abnormal chromosome count, right? So this could be 45 chromosomes, 45 and a half. There could be a failed pair of homologous chromosomes that don't separate in meiosis 1. There could be a failed pair of sister chromatids that don't separate during meiosis 2. What happens is this is a result of this process called non-disjunction. Okay, non-disjunction, an abnormal chromosomal count as a result of non-disjunction, which essentially means that this can change the number of chromosomes in the body. So here's a checkpoint for you. Explain how non-disjunction could result in a diploid gamete. Okay, explain how non-disjunction could result in a diploid gamete. And this could typically be if all the chromosomes are basically affected during one of the meiotic divisions, right? If it's one of the meiosis 1, meiosis 2, where some of the gametes don't undergo a complete separation, you're going to have some accidents. You're going to have some chromosomal non-disjunction, right? Again, an abnormal amount of chromosomes are going to basically be divided, and this is going to result in an abnormal series of resultant chromosomes. So here is non-disjunction in an overall picture. So the first thing you start to see is kind of like that separation should happen, right? But instead of separating, both of those chromosomes go to one of the poles, and as a result, you're going to get a normal meiotic pair of cells, and this one is not going to separate at all because if you notice, there's just one chromosome here separating with two sister chromatids. Here you start to see that separation, but it's separating 6 times, right? So you get 3 separations with 6 individual sister chromatids. So again, this is in meiosis 1 and meiosis 2. And the event you get that, there's an abnormal number of gametes, right? This is n plus 1 and n plus 2. It should n plus 1 and n plus 1. It should be what? Just n, right? This is n minus 1 and n minus 1. Again, it should just be what? 1n and 1n. So in the end, it should be of 1n, 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 1n haploid chromosomes, which are genetically different from one another. But because of non-disjunction, it's not just splitting, especially in meiosis 1 and meiosis 2, they're going to separate to 4 abnormal gametes, which is obviously not going to create a normal egg and ovary, which isn't going to, if it is inseminated, produce a child, right? And so you have issues with that. In normal meiosis, basically, they start to separate, but in non-disjunction during meiosis 2, you get that abnormal pair of gametes, right? So this is going to create a chromosomal difference in the number of overall chromosomes you have in the body. So normally what someone does is, in the first trimester of a woman's pregnancy, usually they do a series of amniocentesis. They take a look at the fluid of the amniotic sac. They determine the blood of the mother if it's actually the same or consistent with what she is before the child's born. And they, of course, look at the blood of the child as well if needed, right? But then they're going to pull blood cells from the actual, the umbilical cord to determine if there's any issues with that, right? All of these are almost standardized tests that they do. You don't have to worry about it. One of the other things they also do to determine the inventory of how many chromosomes you have is do a karyotype. So you remember the lines I drew on the board, the first 22 lines? Those guys were autosomal body pairs. The last one was a chromosome that was specifically XX or XY. That's your sexual chromosomes, right? Those are the ones that are your gametes. That's the 23rd pair. That determines your sex. So that between 1 to 22 and the 23rd is all 23 pairs, which produces your karyotype, okay? So to prepare a karyotype from the body, you just take the patient, you take the baby, you take something. You take the animal, you basically pull its white blood cells. The white blood cells are your ones responsible for immunogenicity, right? Your lymphatic system, really what gives you the healthy overall immune lymphatic system of the body, right? These guys are isolated. We stimulate them. They're going to grow. They'll stop. They'll stop its overall growth at metaphase, and then they're going to take a look at the photograph of what the cells look like under metaphase, because you could determine if there's any chromosomal non-disjunction, right? Once the chromosomes are arranged in ordered pairs, like I said, 1 to 22 or the 23rd pair, any of the chromosomal abnormalities can be easily detected through that event. So again, before you address any type of karyotyping, you got to determine if that patient has a direct number of chromosomes lined up. You got a cell cycle arrested at metaphase and essentially look to see if there's any non-disjunction. Okay, so this is a very important event. There it is. There's the original blood sample from the patient, just standard. You centrifuge it and you separate the plasma from the white blood cells and the red blood cells, which are the formed elements down below. So that's why you guys see the red down below. The fluid is basically the plasma. You separate it. You add something like a hypotonic solution to fixate the cells. When you fixate the cells, you pull the white blood cells from it. The white blood cells are stained. Then you could take a look at it under metaphase and arrest it at that point and determine. So there it is under a very large microscope for a scanning electron microscope or a transmission electron microscope. So you see the physician essentially doing a karyotype. And now you get to line them up, right? 1 to 22, and the last one down here, if it wasn't cut off, should be your 23rd pair. We're going to look at the picture of karyotypes in just a second. So here we go. There's a chromosome with all of its genes and the loci and all the alleles that code for anything from, my favorite, like you guys say, is attached earlobes or mid-digital hair or ability to curl your tongue or brown hair, brown eyes, whatever the color is, right? So all of that is eventually your chromosomes lined up. You notice that the reason why they line them up is because of the banding patterns. So you're really just playing matching, right? Each of these are the same. Each of these are the same, totally different from one another. Look at the structure. And then there's the last one. Long X chromosome, one Y chromosome, right? This guy is a male. So there's your pair of homologous chromosomes. All of the loci are connected. You could do this banding pattern. These guys get paid 6 figures. It's good fun. You could karyotype the individual, and you can essentially determine sexuality, right? Very, very easily, especially when you wanted to figure out if you had a boy or girl. Obviously, in the first trimester, when you do have the penis, and then, of course, you do have no penis, you could determine immediately there, especially with an ultrasound of the cervix, what you have, right? The male or the female. So we got a checkpoint. How would the karyotype of a human female differ from the male karyotype in figure 819? All right, so we got the 23rd pair, right? So the female will have what? X and an X. So 2 of these. And obviously the male is just as is. So again, instead of an XY combination, you're just going to have an XX homologous pair of chromosomes. Keep going along. This is very important, guys. In the 21st pair of a chromosome, there's something called triple X syndrome, okay? That's when you have 3 chromosomes instead of 2. If you have 3, then that child will have Down syndrome, also called trisomy 21. This is actually the most common chromosomal abnormality, and this is the extra copy, again, of that X chromosome that basically looks like it's got 3 in there. So when you do type them and you're pulling them together and you're pushing the chromosomes together and matching them up based on their loci and their alleles, if you got a 21st pair that looks like 3 instead of 2, you have trisomy 21. That's again going to be consistent of Down syndrome. So I had an aunt with Down syndrome. She lived until she was 45, 47. one of those ages. She was in Pakistan. Her caretaker was my other aunt. When my other aunt died of hepatitis, then unfortunately she died shortly later. So children can grow into adults with Down syndrome, right? If they're obviously have that Down syndrome in their 21st chromosome, there it is, one, two, three, instead of just two. What happens is they will live a long life and be perfectly healthy adults, but the problem is there will be an issue with their caretakers. So if there is problems later on where there's no moral support, physical support, mental support, educational support, there has to be that added work. A lot of people say, my Down's brother or sister, they live perfectly healthy lives and they are loving and caring like any other brother or sister I have, right? But there is a time frame where they will not live longer lives than the rest of us. And, we try not to think about that. I obviously wanted my aunt to live in the 70s and 80s. I want all my brothers and sisters to live very long lives, right? But at the same time, it's one of those things that it's more of a physical, mental support. It's the same thing as if we didn't have a roof over our heads, right? Or if we didn't have the correct mental support or the physical support or the correct drugs and the doctors, right, to help us in recovery. It's that kind of support which unfortunately limits Down syndrome individuals from not having the long life that a normal, quote unquote, average American male or female would typically have. Okay, so there it is, the 21st chromosome with 3 in there. That's triple X chromosomes, also called trisomy 21 or Down syndrome. So this is a chart that always freaks me out, right? Because I'm 38, so I'm right here, okay? The age of the mother means a lot because if the age of the mother is higher than in her early 20s, late 20s, 30s, once you hit the 40 mark, there's a super large increase. Even when you get to 45 to 50, there's a large increase that you could potentially have an infant with Down syndrome, right? So there's this age-specific risk related in the amount of live births, and this is only in whites, so Anglo-Saxon Americans and other races from Ohio and metropolitan Atlanta. So total random locations, right? 1998 was a study. We obviously have to take a look at someone from 2018. You guys are more than welcome to, if you're reading this, if you're listening to this document, share the news. Go ahead and write extra credit paper on the amount of individuals with trisomy 21. 1 to 2 pages, like I said, with just a couple of PubMed sources. Tell me what the latest data is, right? Is this still accurate in 2018? Does this still make sense that the number of live births will go to 80 per 1,000 births by the time you're 50, according to this chart? So I'm going to be 40 in about 20 months. So 10 out of 1,000 live births, is that still true? 10 out of 1,000 live births will actually, if that patient is 40 years of age, will still have Down syndrome. So that's something that I look at too, Any aging woman who's, probably pushed on to do other things or gotten married late like I will do, will that actually make a long-term impact on her choice to be a mother, right? Or not. And there's absolutely nothing wrong with not, having children. It's completely up to the individual. I know so many people, happily divorced women who are same age as me, but never had kids either. So there's that as well. There's many sides of the coin that you should take a look at. Happiness is a relative thing. Everyone should try to not compare what quote unquote getting married with 2.5 kids and having a white picket fence is the idea. If, living in an apartment and, having a steady partner and a love interest or something is your bag, then, make it your bag. Don't let society push you to assume that should have children or, propagate your species. My mom had 6 kids. I got 4 nieces and nephews. I'm kind of happy with that, right? And then again, once you get married, you may actually try it, right? It is a long process. It's aggravating. For those people who go through IVF, it's very expensive as well. Insurance may not actually cover it. But you want to try to see if you can actually get to a point where you feel comfortable to eventually have kids. And if it's later on in life, that's your bag, then that's your bag. All right. Okay, so here's some abnormal kind of numbers. Abnormal numbers of sex chromosomes don't usually affect survival. So non-disjunction of sex chromosomes during meiosis can result in individuals with a missing or extra X or Y chromosome. In some cases, that's XXY. This leads to a syndrome that can affect the health. In other cases, it's XXX, where the body is normal. So just because the patient is, XXY for female, that's the third chromosome, X, that doesn't mean that's going to affect the first two. Because the first one's dominated, the second one's recessive, the third one's actually mass. So it's not even a problem where it's actually expressed. So the XXY, that could lead to some effect of the individual, but an XXX can be perfectly normal. So here's some interesting ones. Oh, I mentioned Turner syndrome in class as XO. Klinefelter syndrome is XXY. This is when you have meiosis in the egg or sperm formation. The problem is the individual is sterile and you have underdeveloped testes, secondary female characteristics with an XXY. Okay, so technically you're supposed to be a male, but you have underdeveloped testes, you're sterile, you can't have kids, and you have secondary female characteristics because you have two X's instead of a Y. With Turner syndrome, as an XO, you are sterile and you have immature sex organs. So as an XO, you're significantly female though, okay? For an XYY, you are a normal male and you don't have any symptoms. For an XXX, you are normal female and you have meiosis in the egg or sperm prediction and you're slightly taller than average. So again, very interesting type of chromosomes that can result as a result of an extra a separation or non-disjunction event that happens in the 23rd pair. Okay, so new species can arise from errors in cell division. Non-disjunction can produce poly meaning many, ploid, many different types of organisms with extra sets of chromosomes. Such errors in cell divisions can be important in the evolution of new species. So if you got a polyploid organism, right, this can result in new different types of species. So here we got an example of a gray tree frog, which is a tetraploid organism, which means that if it undergoes different types of unique polyploid extra sets of chromosomes, you can actually create a totally different types of species, which is pretty cool. Okay, alterations of chromosome structures can cause birth defects and cancer. Chromosome breakage can lead to rearrangement. You can delete an entire chromosome. It could duplicate, it can invert, and it can translocate, which means change its actual order. Those changes can produce genetic diseases, or if the changes occur in somatic cells, it could produce cancer. So if you have a chromosomal deletion or a chromosomal duplication, it can result in cancer. So how is reciprocal translocation different from crossing over? This is basically when you swap chromosomal segments, and this can actually relate to crossing over problems. And so if it's between non-homologous chromosomes, that can result in a type of swap, okay? All right, so here we take a look at all of them. Here's the gene, the homologous chromosome, the individual sister chromatid. It's deleted. Now you got this. Duplication is you're duplicating it twice. Inversion is if it's flipping over, right, from green to purple, flipping over, green becomes purple, purple becomes green. And this is reciprocal translocation, where here, this one translates here, and this line translates here. So now you have a unique combination. So again, these are the ones that can result in chromosomal, whoops, we just totally messed it up, types of conversion. All right, so if you take a look at all of them together, you can relate to which one's inversion, reciprocal translocation. Just know that these are the events that can create cancer and can also create abnormalities or a stillborn birth. Okay, if you have the 9th and the 22nd chromosome, reciprocal translocation means that you replace the 22nd with the 9th and flip it. This can activate a cancer-causing gene, okay? So for example, leukemia, which is a blood cancer, this can create chronic leukemia in the 9th and the 22nd chromosome. So those folks who study karyotypes know if things are swapped, then that can actually produce leukemia in the patient. So here's some last minute things you guys should be able to do. Compare parent-offspring relationship in sexual and asexual reproduction. Explain why cell division is essential for both prokaryotic, eukaryotic life cells. And then again, just go through all of them. Be able to explain that. That should be one way for you to know that chapter 8 is done. Okay, so do this with one of the other fellow lab mates or lecture lab mates and see if you guys can talk to each other about this stuff. So start with this before you guys meet up, and it will make a little bit hopefully more sense. These are just some folks that are responsible for understanding cellular reproduction and our Mendelian genetics. So we got the crime scene guy, the forensic scientist. We have the microbiologist who's pulling the information and studying bacterial viral nature. We got the copywriter who is writing everything down from what everyone's learning. So again, the microbiologist studying in the lab. This is the medical copywriter. And then, of course, we have the cell cycle. Here we have the mitosis and meiosis, especially with our gametes of egg and sperm. And now I want you guys to fill this out. This is great so you can identify what the difference between mitosis and meiosis is. And then the last one is just basically anaphase. You can see here an anaphase. This one is in metaphase. Most of these are usually here. We have two cells here. These are mostly in prophase, early prophase or interphase, right? Here you're starting to see separation. But basically try to identify these guys and test your knowledge here in this question. Okay, so chapter 8's finished. Let's go ahead and continue with chapter 9, because we got the video up. We got it rolling. Well, let me just dive directly into chapter 9. All right, so we covered probably at least the first 40, 50 slides, right? Okay, cool. So we talked about genetics, and so let's blow through the parts that we already covered. So we talked about these already, the Mendelian laws, law of independent assortment, law of segregation. We started talking about monohybrid and dihybrid crosses. We were able to cross it with purple and white flowers. All right, and we did the pea experiment with round and yellow and wrinkled and green peas. Okay. Then we talked about chocolate labs. That's right. Black and coded, black and normal vision Labrador retrievers with these cute chocolate labs that are blind, right? So it was a 9:3 to 3:1 ratio. So definitely see if you can perform a test cross and do that. And then we did this experiment with the coins. Okay. Most of us started with this, or the other lab, the other lecture actually started a little bit farther than this. So we've got about 100 slides. We can do this. Okay. So when you talk about pedigrees are basically if I can determine what my family history is, that can help me determine, let's say if Libby has a kid, what's the chances that she can actually determine the kid's genotype, right? So if the kid is, let's say, straight hairline, and we'll talk about straight hairline. Straight hairline is the individual on the left-hand side. Widow's peak is the individual on the right-hand side. If she's got a kid with a straight hairline, Libby would know herself that she's heterozygous, right? So if we get the straight hairline, which is two lowercase h's here, and we got a widow's peak, that means it's capital H or a lowercase h, then basically the mom, Libby, would be heterozygous as well. Because the straight hairline is two lowercase h's, the mom's got to be one of the h's, so she's going to be capital H and lowercase h, which is a heterozygous trait. So here we have the individual. Let's see, we got Al and Beth. Al is a male, he's a square. Beth is circle. So anything that's square is a male, anything that's circle is a female. So you see that in mating, Al and Beth will have 4 kids. Charles and Debbie are going to have 2 kids, right? After this, Isabel and Henry are going to mate, and those guys are going to have Kristen and Libby, right? So Libby, as you noticed, is got a circle with a open area. So she's a female with a widow's peak, and you can tell that. Here, Kristen is a female with a straight hairline, so you can identify that. The question is, both Henry and Isabel were what? Isabel had a widow's peak, and Henry had a widow's peak. So that meant one of them had a widow's peak. Okay. Here we start to see Al and Beth. Both of them are normal, but then all of a sudden, 2 out of 4 of them end up having straight hairlines. That's why it's still in the lineage for Kristen to have a straight hairline, right? Charles and Debbie are getting married over here. One of them has a widow's peak. Both of them end up with both widow's peak having one with a widow peak. So we're going to take a look at a parent, the parent Henry and Isabel. The parents both have widow's peak, right? And they have a child who has to be a straight hairline. So in the generation, right? So here we have straight hairline. In this case, the parents are heterozygous, both here. That means the straight hairline has got to exist. That's why Kristen has, in this case, a straight hairline, but her parents don't, right? Her parents don't. Her parents were widow's peaks. So Kristen has a straight hairline, but her parents don't. So the straight hairline must be a homozygous recessive trait, okay? Down here, not all genotypes can be determined. Libby could be what? Libby could be a dominant heterozygous dominant widow's peak, or she could be, sorry, not homozygous dominant, or she could be heterozygous. But that's really unknown because unless she has some kids, we don't know exactly what her genotype is, okay? So a lot of genetic disorders can happen, right? It depends on if the parents are carriers, right? If it is true that both of them are carriers, they're probably heterozygotes, which means that it's a capital H or lowercase h. But the parents who are carriers will be the carriers for the recessive allele, but they'll be phenotypically normal, right? So what's the odds, let's say down here, of having these type of autosomal disorders in humans? Here's some examples, by the way. Albinoism, that's basically if you have a lack of pigmentation in the skin, you have light blue eyes, you have white skin, right? White hair, okay? Cystic fibrosis is another type of autosomal recessive trait. It is extra mucus in the lungs, digestive tract, liver, increased susceptibility to infectious diseases, and unfortunately death in early childhood unless it's treated. PKU, or phenylketonuria, is an accumulation of phenylalanine in the blood, a lack of normal skin pigmentation, and developmental disabilities later on. Sickle cell anemia, which I'm sure most of you guys have probably heard of, is typically among African American individuals. Those are sickle cell shaped type blood cells. They kind of look like moons, right? And it can damage many of the tissues. There's tired, lethargic behavior. They can live normal lives, but they typically will have a lot of just lethargy throughout because you don't have much oxygen delivered to the tissues simply because of the sickle cell shape of the red blood cells. Tay-Sachs. Tay-Sachs disease is lipid accumulation in the brain, mental deficiency, blindness, and death in the child. And then we have some dominant disorders. They're autosomal, but they're dominant. Achondroplasia, which is dwarfism. Mini-me, right? Everyone's watched Game of Thrones, right? Dwarfism is a type of achondroplasia, which is a dominant disorder. Huntington's disease is actually a kind of blood-developed disease. It's a developmental disease that you have a lack of control of your movements, and you have some cognitive impairments, and unfortunately it strikes kind of in your 30s, okay? Hypercholesteremia, excess cholesterol in the blood. That's totally me. My entire lineage, no matter who I marry, I will have that hypercholesteremia, dominant autosomal disorder. That's just increased cholesterol. My cholesterol's through the roof. I try to eat healthy, but I can't really do anything about it simply because I have so much cholesterol lining my arteries and veins, which means I have atherosclerosis. Okay, checkpoint. Peter's 30-year-old. His father died of Huntington's. Neither his mother nor his older sister show any signs. What's the probability that Peter's got it inherited? It's got to be half a chance, right? The genotype of the sister is irrelevant. So the father had the disease. There's half the chance that Peter will receive the disease, okay? All right, let's take a look at more things. These are the kind of characteristics of some of the dominant traits. Freckles. Recessive traits is known for freckles. Normal pigmentation, albinoism, right? Again, freckles, no freckles, normal pigmentation, albinism. Here is the parents. The parents, if they're normal, that means they're heterozygous. If they're heterozygous, you split the sperm, which is the male. Whoops. Here's a karyotype we talked about again. Oh man, it just split. So we split the sperm. It's heterozygous, A and lowercase a. Heterozygous again for the female. We put her over here, A to A. We do the cross. This is homozygous dominant. This is heterozygous, and this is homozygous recessive. So you get a 1:1 ratio, right? The albinoism is that recessive disorder. So that's why there's a 1 out of 4 or quarter chance ratio that you can become an albino if you have it as a recessive trait. We got a person here doing an amniocentesis. Actually, my bad. You got the surgeon here. My bad. I just noticed it right now. He's got achondroplasia. So this is basically a surgeon who clearly can live a long, healthy, happy life and become a surgeon. How amazing is that? That's awesome. So it's not like you can actually sort of stop an individual with a defective disorder from having a long, healthy, productive life. These, they'll be perfectly healthy and live long lives. So new technologies can actually provide insight into genetic testing. For example, carrier testing, fetal imaging, newborn screening, amniocentesis, right? Or chorionic villus sampling, CVS sampling. Chorionic villus sampling is basically a genetic screening where I take the chorion, which is part of the entire sac, around the amnion, and I pull some fluid out. And what I'm going to do is, in early pregnancies, I'm going to figure out if I've got any chances of miscarriage. Of any chances of genetic screening. But the problem is, if you do a CVS sampling, it'll carry a risk of miscarriage because you're kind of working with the unborn child that's obviously still inside in utero. And if you pull a little bit out of that amniotic sac, right, the chorionic villus sampling could potentially, there is a small chance that it could carry a risk of miscarriage. So here is the cute little fetus sitting inside the cervix. We have the placental wall, and then obviously we have the vagina, which is going to be external genitalia, and the birth canal, right? If you do an amniocentesis, you essentially extract fluid out of the amniotic sac, right? While you're transducing it with an ultrasound, you gather basically with that extraction, so a lot of the fetal cells, right? After a while, to several weeks of testing, you can determine karyotype, right? So you're going to take a couple weeks to figure out the karyotype, to figure out if you have any abnormalities, right? If you do CVS sampling and the fetus is much smaller, right? You have to try to suction the tissue and pull it, because then you do the sampling and the needle goes through here and you have to pull directly the fetal cells from the fetus. And because you're actually kind of disturbing the fetus, and that could actually lead to some potential chances for a miscarriage. So it's kind of scary when you do CVS sampling, right? But the good positive thing is, in a couple of hours, you could do a karyotype and along with the genetic testing and get your results faster. So it depends, and if, let's say, you have a damage to the fetus and it's very timely when you do the sampling. Amniocentesis are kind of normal. You want to try not to do it as much as possible, but if it's needed, do it, right? It's kind of commonplace these days. CVS sampling is not done as often, but in the case of, let's say, damage to the cervix, damage to the fetus, there's abnormal difficulty, there's difficulty with the pregnancy, you can actually obviously try to suction some tissue from the chorionic villi and gather those fetal cells, but it will be some risk of miscarriage, but you'll still get that karyotype in a couple of hours as opposed to, several weeks later on with just the amniocentesis. Okay, this is a traditional ultrasound. We've got happy mom taking a look at the ultrasound, and so she could see images that you could determine, in this case, heart waves. You could determine sex of the individual, and it's actually pretty cool, right? Let's take a look at, where's my volume? Where's the ultrasound? Where's my volume? Ah, I didn't get any volume, but you saw the infant, the fetus still moving. Looked like he was kind of suckling his fingers. At that point, I kind of lost imaging idea. I obviously haven't had any kids, so I don't know exactly what each of the individual movements were, but this is the amniotic sac, right? That's where you're going to pull the amniotic fluid from, because the chorionic villus sampling would just be a sample of this chorion, right? And it could disturb the fetus. So there's a lot of things that can happen, right? Okay, so fingers crossed, hopefully even if you do have a CVS, things will be well. There is some differences, though, when we talk about Mendel's laws. Mendelian genetics basically looks at two things. Can something be or have complete dominance, or can it have incomplete dominance? Incomplete dominance means that you're kind of getting intermediate phenotypes. So let's say you got a filial 1 hybrid, and it falls between the phenotypes and physical appearances of the parents. That's called incomplete dominance. If you have, let's say, with Mendel's P's, if you cross them and one of the parental varieties, it always looks like one of the parental varieties, you got a situation of complete dominance, right? So Mendel's laws are really valid for everything. You can do it for Peas crossing, you can do it for humans, you can do it for Drosophila melanogaster, you could do it for musculosis, you could do it for the macaque monkeys, you could do it for anything, okay? All right, so we get a red and white flower. Red is dominant, white is recessive. So this is homozygous dominant, homozygous recessive. If they have the filial 1 generation, the first set of kids, you're going to have a hybrid of a heterozygous. The heterozygous is going to do half of the dominant, half of the recessive. So this next generation is going to be half of both each of the dominant and recessive. Same for the eggs. You're going to have a 1:2 to 1 ratio of red to pink to white flower. So again, you have the red and white mom and dad. Here we have the hybrid heterozygous pink flower, and essentially the grandkids will basically be 1:2 to 1 ratio of red to pink to white flower. So why doesn't this cross shown export the blending hypothesis? Remember that? Because although the two kids here are pink, they got blended, the other are not, right? So the white and the red alleles are not lost to future generations. So you can't just continue to blend them all pink. You're still going to have a 1:2 to 1 ratio. So that's why the blending hypothesis does not work. You're not going to always get like a blend between two things and pinks all later on. No, you're going to get a 1:2 to 1 ratio. You're still going to see some dominant pink, and then you're going to see some recessive purple and white. Okay. Let's talk about LDL receptors, low density lipoproteins. So if anyone's ever taken their cholesterol, there's two things, HDL and LDL. HDL is your good fat. LDL is your bad fats, right? Low density lipoproteins. So the normal thing is if you have something homozygous, you have something homozygous dominant, it's two H's. It's heterozygous, it's mixed H, dominant and recessive. Homozygous recessive basically means you don't have an ability to make LDL receptors, right? That's bad. So if you're homozygous recessive and you can't make LDL receptors, your body can't uptake all of those fats, whether they are good fats like your olive oils, right? Your extra virgin olive oil, your sunflower oil, right? Not your butters, because butters are LDLs. If you have some mild disease, some can uptake your LDL, right? So this is here an example of hypercholesteremia, which is increased cholesterol inside the body. If you don't have those receptors, like here I'd probably, whoa, where did it go? Yeah. So here I'm probably some mild diseased where I have just a heterozygous genotype for LDL receptors. So half of them are uptake and the other ones aren't. So this causes some mild malformation. I actually have a cholesterol of like 270, which is awful. I should actually be on a statin. 4 out of my 6 brothers and sisters are on statins. I'm the only one, along with my healthiest sister. She doesn't need to be on any, that are not on statins. I need to be on a statin. I've asked for statins many times. This should lower the cholesterol in my body. But the doctors are like, Professor, just exercise and diet. I'm like, good God, just help me. You know? So the thing is, though, if I have a kid, I shouldn't have hypercholesteremia, because that will then give you in utero childhood diabetes, right? So which is going to cause problems during the birth. So you should typically be on a cholesterol lowering medication to try to make sure you're not going to get a heart attack or a stroke, mostly a heart attack. And if it's a case where you just don't have the receptors and you're homozygous recessive, you're probably going to be severely diseased and, kind of have a stroke and heart attack early on in life, which is really, really sad, but that's really one of the things that could happen. Okay, let's talk about blood types. Blood types usually come in A positive, A positive, A minus, A negative, B positive, B minus, O positive, O negative, right? So that's part of the ABO blood type. There's 3 alleles, A, B, and O. And you can have AB positive and AB negative, right? So most of the alleles are co-dominant. That means both of them are expressed individually, A and B. So if you have alleles for both AB blood, you have IA and IB alleles. They're co-dominant with one another, okay? So here's a checkpoint. Maria's got a type O blood. And by the way, O negative is a universal donor, okay? And her sister has a type AB. By the way, AB positive is a universal recipient, okay? The girls know that both of their maternal grandparents are type A. What are the genotypes of the girls' parents? The mom's got to be what? Homozygous for type A, so she's IA, and the dad's got to be homozygous for type B, that's IB. So that's the genotypes of the girl's parents, IA and IB, right? So here is the breakdown of all of her blood types, A, B, AB, and O, okay? A, B, AB, and O. The genotypes are listed. This is A, this is B, this is AB, and this is O. If you have carbohydrates present on the red blood cells, you'll have sugars for A, sugars for B, sugars for A and B, and none. Your antibodies. So you will produce naturally, and you'll memorize this later on anyway. But if you have antibodies for type A blood, you have anti-B. For type B blood is anti-A. For antibodies for AB, you have none. That's why you're the universal universal recipient. And then type O, O negative specifically, you're the universal donor, right? We love you guys because you basically can donate to everyone. So this is what happens. If you have clumping, that means that there's an interaction. It's bad, right? Reaction when blood groups below is mixed with antibodies from blood groups out left. So if you have blood type A, you're going to be fine if you get blood from O and A. You're going to be bad if you get blood from A, B, and B. If you're type B, you can get blood from B and O, right? If you're type AB, you can get blood from everyone. You're the universal recipient, okay? If you're type O blood, you can only get blood from type O, but you can donate to everyone, and every other blood type will make you clump and coagulate. Okay, so this is basically AB, which is universal recipient, right here. Universal donor is O. Type B blood can get blood from B and O. Type A blood can get blood from A and O. Okay, so that's your multiple alleles for your ABO blood type. Let's say you have a gene with many phenotypes. This is called pleiotropy. Pleiotropy occurs when a gene influences multiple characteristics. So, for example, sickle cell anemia. Sickle cell anemia gives you that characteristic sickle cell shaped red blood cells. This affects the amount of blood protein called hemoglobin that will attach to oxygens. So if you have less oxygen attaching to your hemoglobin, basically what that means is you can't transport oxygen to your tissues enough. You'll basically have 3 oxygens to 4 oxygens being transported. Then the tissues get the oxygens and they're like, okay, that's 3 out of 4. Where's the rest? I'm just going to get tired in the meantime. So they're waiting for the other quarter of that oxygen. They got to go run through the lungs again, back to the heart, dispense it through the tissues. The tissues are like, okay, well, thanks. I got 3 out of 4 oxygens again. Basically, you become hypoxic, which means that you will essentially lose that oxygen. You'll become lethargic, tired, weak, and that's what the result of sickle cell anemia is, okay? All right. So sickle cell disease affects folks with hemoglobin. Those are the carriers of the disease. Typically, they have increased resistance to malaria as well. Malaria is a disease that is spread by, it's a viral disease that is spread by the Anopheles mosquito. So certain mosquitoes can transmit malaria, and folks with malaria will have fever, vomiting, some flu-like symptoms for several weeks, not normal to a week that we have it or a couple days that we have it. And those long sicknesses can potentially, or unfortunately, to be quite honest, cause some death. But malaria is one of those viruses that we're hoping to try to create a vaccine for it. The Bill and Melinda Gates Foundation is trying to do that. And right now they're trying to give the vaccine or develop something to prevent malaria. One of the big thing is just get mosquito nets, insect repellent. You know, you standing water, take the water and take it out of the pan. So you don't have a place for the mosquitoes to drop their larvae or their eggs directly into the water, so more mosquitoes are created, right? So a couple things you can obviously do to deter potential chances for mosquitoes to be in the environment. Okay, why is sickle cell anemia codominant, right? It's because both of the traits are expressed. You got a carrier for the sickle cell, and both are doing abnormal and normal hemoglobin. Here's that sickle cell. This is the abnormal hemoglobin. This is kind of like the jagged edges. These are normal red blood cells. They're biconcave erythrocytes. They're like 5.2 micrometers in diameter. They are basically your capillaries. Each of your blood cells, your erythrocytes, can bend, so they have pliability. So they can basically go through all of your capillaries, right? But they line up one by one because capillaries are the smallest type of vascular elements in the body. All right. So you got a kid. That person's homozygous for sickle cell. He produces an abnormal hemoglobin, sickle cell hemoglobin. The abnormal hemoglobin crystallizes. It causes red blood cells to become sickle-shaped. A couple things can happen. Pain, fever, joint problems, physical weakness, anemia. Talked about those because you got basically hypoxic, right? You could have pneumonia and other infectious diseases as well. The other things you could do is it could damage the organs. Obviously, blood, if it starts to die off, it will essentially go to the spleen and die off there. So you could have spleen damage. You could have kidney failure because the kidney's responsibility is to filter that blood as much as possible. Heart failure, and unfortunately brain damage. So there are some long-term problems with sickle cell anemia as well. A single character may be influenced by many genes. So a polygenic inheritance means that, for example, your skin color is basically polygenic. So there isn't just one phenotype. We're not just all white, right? We're mixtures of all white, brown, black, unique skin color types, right? So you got a kid. He's got 2 heterozygous and 1 homozygous recessive individual. And it's indistinguishable genetically from a phenotype with these individuals, right? This individual would be indistinguishable in phenotype from which one of these, right? And the answer is everyone except this guy right here, okay? Every one of these he'll be indistinguishable from except this guy right here, right? Because each of these has a carrier that's dominating A and B, right? So this is A and B. And this is the only one that it will be indistinguishable from. Okay, so wow, we're getting to some very detailed shades. Okay, homozygous recessive for A, B, and C, very light. Your homozygous dominant, you're very dark. If you have a medium shade, you have these combinations. And here's, whoa, a crazy amount of combinations. This is just telling you the fraction of individuals for this polygenic inheritance of skin color that give you a variant, beautiful array of skin tones. I just think it's one of the most beautiful things in the world. Okay, so light, dark, gorgeous, and then you have these unique, beautiful shades of medium as well. And then you have all of these combinations. I'm not going to repeat them out of 64. Majority is, most of us are going to be mixed, right? Or variances of it, right? And then there's very rare for the other extreme polygenic skin characteristics, okay? So many traits are affected in varying degrees by both genetic and environmental factors. If most characters result from a combination of environmental and heredity, why was Mendel able to ignore environment from his pea plant experiments? So what he did was he basically raised everything in a similar environment. That's why he could ignore environment, okay? Here's our beautiful exposure to the sun. All of it can affect us differently. This individual's got slightly red. This one's definitely pink, and this one looks like it's burnt, right? So you could have had individuals with all the same skin color, but obviously they could be burnt in different ways. Here we have twins. These are genetically identical twins. Apparently the author of the book, these are his daughters, so maybe that allowed him to study his stuff, right? Okay. All right, so let's take a look at the chromosomal inheritance, basis of inheritance. So really this talks about loci, right? certain genes are inherited based upon a chromosomal theory. So we talked about how they'll separate in meiosis. So which one of Mendel's laws, the law of independent assortment and the law of segregation, follows below? So which one is it where the orientation of homologous chromosome pairs up in metaphase 1? That will be the independent assortment law. What about when they separate? If they separate, they are in the law of segregation for B. Okay? So if we have all seeds that are round and yellow, that is, in this case, heterozygous for round and yellow, you notice that in anaphase and metaphase, you're basically going to have a 9:3:3:1 ratio, right? This is the chromosomal basis of Mendel's law. We talked about this already. We don't have to go through it again. We can look at Punnett and Punnett square. So Punnett was one of the individuals who decided to study the 9:3:3:1 ratio. Bateson was the other one. They said that plants didn't show the 9:3:3:1 ratio in F2 though, because there were some linked genes. Though some of the genes were inherited and some of them were close together. So in what ways was Bateson and Punnett success dependent upon failing at first? Because if you didn't really look at the failure of linked genes, you couldn't really discover them, okay? So here we have purple and purple flowers with long pollen. Totally different from purple and white, right? Purple flowers with long pollen. So if we have purple flowers with long pollen, and we look at the generations of phenotypes, you're going to have purple and long majority offspring, and then you're going to have red and round and some purple and round and red and long, right? The prediction was kind of in a 9:3:3:1 ratio, right? The explanation was in meiosis. So when you take that purple and long pollen flower, most of the gametes will be purple and long, but they'll be split 3:1. 3 purple, 1 red. 3 purple long, 1 red round. Okay, so this doesn't really account for a number of things. This really linked what? Genes. This told us that genes were linked together. So again, once we do the experiment, we determined that genes were really linked together. Okay, so another thing we should look at is crossing over. Crossing over between homologous chromosomes produces new combinations of alleles and gametes. So linked genes can be separated by crossing over, and you can get recombination. So this is the recombination, again, from crossing over. You have this tetrad pair of homologous chromosomes. Their entire lower leg of chromosomes cross over, so the gametes will recombine later on. So you see a unique Drosophila melanogaster. We always study this. We know that there's like 130 ATGCs, so it's very small in terms of genetic sequence. So we know everything about our studies mostly from playing with the Drosophila melanogaster, which is our fruit fly. You guys probably studied this guy in high school. Okay, so we have, let's say, gray body with long wings, right? Gray body with long wings with a black body with vestigial short wings, right? If this guy's offspring are created, there's a 17% chance recombination frequency, okay? So this is the role of crossing over and inheritance. The 17% or 0.17% chance of recombination occurs, so you can get any different unique percentages, right? When you take a look at the data in this figure above, what's the recombination frequency between the flower color and the pollen length? And this one should be around 11% for this one. Okay, we're taking a look at data on genetic maps. This tells us where the loci are located on the chromosome, and then we can link it together. So let's take a section of chromosomes with these genes, linked genes of loci of G, C, and I, or L. There's a 17% difference between G and I. There's a 9% recombination frequency between G and C, and there's a 9.5% recombination frequency between C and L, right? Or C and I, whatever that is. So depending upon the location of where they are located, then you can get different percentages of recombination frequencies. If they're closer together, you get a recombination frequency that is 9.5%. They're farther away, you get a greater percentage of a chance of recombination. So if we take all of the short aristae, that must be the little ear thingies, maroon eyes, black bodies, cinnabar eyes, vestigial wings, down curved wings, and brown eyes, with all of these, we can look at the wild type, more common appearances of red, normal wings, gray eyes, gray bodies, red eyes, and we take a look at those guys and determine its frequency. So if you design an experiment to provide recombination data, the gene has recombination frequencies of 3%. With the vestigial wings, and which is this, whoops, oh man, what just happened? With the vestigial wings here and 7% cinnabar I, right? Where's it located on the chromosome? Okay, so the gene is actually between the vestigial wing, this part right here, and the cinnabar loci. And it's a bit closer to the vestigial wing combination because the vestigial wing locus has a lower recombination frequency. Okay, let's take a look at sex-linked chromosomes. What's linked on the X chromosome? This is really important because there's a lot of genes that are directly sex-linked, X-linked chromosomes. So these are basically XX linked, right? So if a male is XY and a female is XX, the Y chromosome has genes for the development of testes, whereas an absence of the Y allows for ovaries to develop, XX, right? In addition, human males and females both have 44 autosomes, non-sex chromosomes, right? Because the other two autosomes, which 46 equal, is your sex chromosomes. So other systems of sex determination exist in other animals and plants, okay? In some animals, environmental temperature determines sex. That's interesting. For some reptiles, the temperature at which eggs are incubated during a specific period determines whether the embryo will develop into a male or female. Global climate change may therefore impact the sex ratio of certain species, right? Unfortunately, Scott Pruitt of the EPA, the moron who runs the EPA, currently said that climate change was not real. Good God help us all, right? We are screwed. All right, God help us all. Okay, so King Henry VIII I am, of England, was quick to blame his wives for bearing him only daughters. How awful. Explain how, from a genetic point of view, this thinking was wrong. Why? Because males are the ones that give you either the X or either the Y. So the males determine the sex. The wives always carry two X's, so the wives will always give an X. It's the crazy men. So gentlemen, quit blaming your wives for giving you another son or daughter. Blame yourselves because you're the ones that give the X or the Y. You know, it's unfortunate. There's a lot of countries out there with certain cultures, even mine included, that love to blame the women for not producing males or male heirs or silly things like that, right? And so they divorce the wives. And so this is, it really just rattles me because how could you not know that the female is the one with the XX and the male is the one with the XY? So the male is the one that only can donate one of the X's and one of the Y's. The female can only donate an X, so the person to match it up would be the male with the X and the Y. So why is it that we still have so much female blame game? It's absolutely unfortunate. Okay, this is sex determination. This is kind of cool. So there's three systems of sex determination. This is the system. We got a grasshopper here, we got a rooster, and we got a bee, right? So we got the genetic makeup of males, which is 22 plus X. Females is 22 plus X. Males here is 76 plus ZZ, and females is 76 plus ZW. Genetic makeup for B is 16, and for females is 32. So it's kind of cool. There's probably different variances of males and female species that produce different types of chromosomes and numbers. So it's definitely different for all those systems. Here's an example of the human sex chromosomes we talked about. There's X. There's that disappearing Y chromosome I talked about earlier. Oh boy, you guys again can write that extra credit paper, more than welcome. Oh, we're talking about cute kids now. Okay, so we got a male. Male is 44 plus XX, excuse me, XY. Female is 44 plus X. XY? Why is it XY? Kind of confused here. Whoop. The male has gametes of 22, yep, plus X and Y. The female, 22 plus Y? Why is this XY? She's XX. Female and male. All right, I'm kind of confused here. Why is this a male? Why is this a female? I'm a little confused here because this is what's throwing me out. This should be X, and this should be X here. All right, I'll see what that picture talks about in just a second. I'll tell you guys later if I figure it out. If not, we're going to read it from the book, okay? All right, good. So sex-linked genes are exhibited, sex-linked are called X-linked genes. So a gene located on a sex chromosome is called a sex-linked gene. The X chromosome carries many X-linked genes that control relates unrelated to sex. So white eye color in the fruit fly is illustrative of a sex-linked X-linked recessive trait. And let's talk about a couple more of them. We got a white-eyed female Drosophila. She's mated with a red-eyed wild-type male. What result do you predict for the numerous offspring? Okay, so we got a fruit fly eye color determined by sex length, right? White-eyed, red-eyed. Okay, we got the female red-eyed and white-eyed. So the female deposits the red, the male is recessive. So here we have a red-eyed allele and a white-eyed allele. So we have the sperm, which obviously is going to produce either the X, R, or the Y. So the sperm is going to produce the difference is if you've got a red or white-eyed baby Drosophila. Okay. If we take a look at the male and female again, right, and we determine the eggs, if we have a recessive and then we heterozygous and then we have a dominant, we're going to determine we have 3 red eyes and 1 white eye. Okay, here we have a combination for 2 red and 2 white. So most X-linked disorders are due to recessive alleles and therefore are seen mostly in males. So sex-linked, X-linked disorders are usually in males, mostly like hemophilia. That's an inability to clot blood. A male receiving a single X-linked recessive allele from his mom will have the disorder. A female must receive the allele from both parents to be affected. That's why it's kind of hard for the female to actually get a sex-linked, X-linked disorder. Okay. Neither Tom nor Sue has hemophilia, but their first son does. Also, color blindness is a sex-linked, X-linked disorder because most males are colorblind. This is just another example. If the couple has a second kid, what's the probability that he or she will have the disease, right? 50%. Yeah. It's half chance the male will have the kid and half the chance that he will inherit the mutant, okay? So this is Henry VIII, Queen Victoria and Albert, my bad. And he so if you want to study hemophilia, which basically is where did hemophilia even come from? We are all related to this, okay? So if in the story, Alexis was the carrier because Queen Victoria, she was a carrier, Albert was not. Then their daughter, Louise, was married to Louis. Alexandra was still a carrier. She married the Tsar Nicholas, so this is the Tsar Nicholas. Tsar Nicholas of Russia, my bad. And he's not a carrier, but Alexandra was. And then Alexis became hemophiliac as a result. So the Y chromosome provides clues about human male evolution. Y chromosomes can provide data about recent human evolutionary history because they are passed on intact from father to son. Okay. Like the Y chromosome, mitochondrial DNA can be used to trace maternal ancestry because mitochondria are characteristically inherited from the egg. It costs a little bit of money, but you can do a genetic ancestry, right? So anyway, back to the Y chromosome. In 2003, geneticists discovered that about 8% of males currently living in Central Asia have a Y chromosomes of striking genetic similarity. Further analysis traced their common genetic heritage to a man living about 1,000 years ago. Guess who too? Genghis Khan. So in combination with historical records, the data led to speculation that Genghis Khan may be responsible for spread of the telltale chromosome to nearly 16 million male descendants, which is crazy that all of it was linked back all the way to Genghis Khan. So here's Genghis Khan. He's a Mongolian ruler. Genghis Khan, I think, killed a bunch of people too. I don't know the history of it. You guys tell me the history, but he was an invader. Like at the time, were invading many lands, but he's known for basically having a sex-linked, X-linked disorder, which according to this, 17 million people eventually are connected to. Okay, here's the end of the entire chapter. Make sure you guys are able to do all of these. Tell me the theories of pangenes. Tell me the difference between P generation, F1, F2. Determine all of this and then explain it to your fellow lab mates. And then you guys are good to go. Have a fantastic Friday, guys. Enjoy your weekend. And I will talk to you guys later.